Photosynthesis is a biological function in bacteria and plants and is carried out by protein complexes called chloroplasts, which harvest light energy and transform it into chemical energy. Photosystem 1 and Photosystem 2 are complexes inside of chloroplasts involved in the primary photochemistry of photosynthesis. Both photosystems are similar because they gather light energy, but they differ in their application and energy level. Organisms may have either one of the two photosystems, or they may have both.
Cyanobacteria are photosynthetic prokaryotes, believed to be the first photoautotrophic organisms to split water molecules into oxygen gas and free protons. These organisms are largely responsible for the great oxygenation event of the early Archean eon. Cyanobacteria carry out photosynthesis very similarly to plants. They have both photosystems 1 and 2 and utilize a non-cyclic electron transport system to produce ATP while simultaneously reducing NADP to NADPH.
A key feature in cyanobacteria that plants do not have in their photosynthetic cycle is phycobilisomes. So what are phycobilisomes? PBS have large protruding rod-like structures that offer a vast surface area for the capture of light. Because of this greater surface area, phycobilisomes are much more effective at gathering light energy than either of the two photosystems alone, and offers its energy mainly to photosystem 2 but sometimes to photosystem 1 if necessary. Phycobilisomes play an important role in cyanobacteria's ability to capture and process light energy in places where light is scarce, such as deep underwater.
Cyanobacteria have the unique ability to rearrange their photosynthetic apparatus to optimize the capture of incoming light. This process mainly involves the expression of different genes that replace or remodel proteins in the phycobilisomes and photosystems. Generic pigmentation is replaced with more functionally distinct pigments and diverse phycobiliproteins that absorb certain wavelengths of light more efficiently. This process is known as complementary chromatic acclimation, or photoacclimation.
Leptolingbia, strain JSC1, is a unique cyanobacterium that possesses an exceptionally advanced form of photoacclimation called far-red light photoacclimation. JSC1 radically changes expression of almost half of its genome to overhaul the photosystems and phycobilisomes to operate in wavelengths greater than 700 nanometers. This reconstruction results from the transcription of a special gene cluster that codes for special far-red pigments.
In a certain case study, JSC1 was grown under 5 different radiation conditions: normal white light, green filtered light, red filtered 645 nanometer light, 710 nanometer light, and far red light. The cyanobacteria exhibit no absorption for wavelengths of light past 700 nanometer for the first 3 light sources. For cells grown in 710 nanometer and far red light, absorptions at wavelengths below 670 nanometer were drastically reduced, but uptake for wavelengths from 700 nanometer to 800 nanometer had a threefold increase over cells grown in other cell sources. This pattern is indicative of special pigment synthesis.
This graph shows how different genes in the complexes of cells grown in far red light have different transcription rates. Transcript levels were observed to have grown for cells grown in far red light, while transcript levels for genes unassociated with far red light decreased. During this far red acclimation, most core polypeptides of photosystem 1, 2, and phycobilisomes should be replaced by products of the 21 gene cluster to optimize far red light absorption. To verify that this was indeed the case, photosystem 1 and 2 complexes were remodeled under far red light. Proteins of photosynthetic complexes isolated from JSC1 cells grown in white light, far red light, and 645 nanometer light and 710 nanometer light were analyzed. Figure A shows reduced activity in photosystem 1 for 710 and far red light compared to white light and 645 light, indicating an increased activity in the phycobilisomes of the cells, which are crucial for photoacclimation. Spectra observed in figure B indicates that both photosystem 1 and 2 complexes of cells grown in far red light or 710 light contain chlorophyll F, chlorophyll produced during photoacclimation, which absorbs further than any other chlorophyll in far red light. The core polypeptides in the cells grown in far red light or 710 light were the products of genes whose transcript levels strongly increased in cells grown under far red light, while the cells grown in white light or 645 light contain products of genes with lower transcript levels under far red light. This indicated the expression of different genes that replace or remodel proteins in the phycobilisomes and photosystems. Figure A and Figure B visualize the replacement of core subunits of JSC1, including photosystems 1, 2, and the proteins of the phycobilisome cores working to make absorption of far red light more efficient.